everybody, welcome to the ARD Simplified Series going on for the NABARD Grade A examination. And the topic that we have picked up today, it is breeding in livestock. And why have we picked up this one topic only? Because if you look at one of the previous year, there was a descriptive question that was asked on this topic. What is cross breeding? And explain the different methods of cross breeding with the advantages and disadvantages, right? So that is why we have decided to talk about or discuss about breedings in livestock and see cross breeding is a part of it. So we will start off with understanding like what is breeding, what are the different types of breeding, and then we are going to look at specifically what are the different types of cross breeding, and at the end we will also talk about the modern ways of breeding culture that is currently going on in the world, the modern techniques that are there. Okay, so this is going to be the flow. So starting off with why the breeding is done. So it's the concept is very simple. That is, we want superior genes in our progeny, so that for whatever purpose are we rearing those animals. It can be cattle, it can be goat, sheep, anything. So whatever we are rearing it for, it can be for meat, it can be for milk, it can be for the wool that they produce, or anything. For whatever purpose that we are rearing them, we get the best quality for it. Okay, so that is why breeding is done. So limiting the reproduction of inferior animals means animals with inferior genes to crossbreed them with other such breeds, so that we get the superior animals in the progeny. And sometimes it happens, not always. It's like, there are inferior animals and they are being mated with the superior ones to get better progeny. Sometimes both the parents are of superior quality only, so that in the progeny we have best of both worlds, you can say. Okay, so why is it done? So that we get all in all, if you have to say, breeding is done so that we can have superior progeny with the desirable genes that the breeder or maybe the farmer wants to have. Okay, and it depends on for what purpose are you rearing that particular animal. Okay, so next generation में हमको मिले. Broadly we can classify breeding into two types. One is known as inbreeding. We call it inbreeding when the mating is done among the relatives. Relatives means those animals that we are using as a male, as a female for crossing, they are somehow related. If they are related, they have some commonality in genes, that is known as inbreeding. Another one is outbreeding. In outbreeding, the animals, they are not related, as simple as that. Okay, inbreeding can be of further two type: close breeding and line breeding. What is the similarity and difference? Because both are inbreeding, so definitely in both relatives are used. Okay, but in close breeding, immediate relatives are used. For example, mating a dam. What is a dam? The cow, which is a mother, that is known as a dam, and son. Okay, so means the mother and son, when the mating is between mother and son, now they are the immediate relatives. They share the same genes, or you can say 50% of the genes, right? Because mother genes must have transferred in the son, so they are the immediate relatives, and mating is done between them. Sire is the male parent, that is the sire and daughter. So when father and daughter are mated, that is the close breeding. So means it is between relative, full brother, full sister, and they are immediately related to each other. While in line breeding also relatives are used, but not that immediate. For example, grandson and for example, mating of grandson and granddaughter, or grandson and granddam. For example, it was grandsire. Okay, grandsire, and then from grandsire, and then you must have sire, and then it must be for example daughter. Okay, so in case of close breeding, sire and daughters were used, right? That were the those were the immediate relatives. But in case of line breeding, grand sire and daughter is used. So you can say that one progeny is skipped. So in line breeding, what happens? Breeding is among the relatives only. Definitely from this grandsire, some gene must have transferred into the daughter, right? With the lineage, right? Definitely it must have done, but they are not immediately related. So breeding is of two type: inbreeding, outbreeding. In inbreeding, relatives are used. In outbreeding there are no relatives. They can be of same breed but they are not relatives. ठीक है. Inbreeding में of course they will be of same breed. Of course if mother is for example Sahiwal, then the daughter will also be Sahiwal, right? So that's common understanding. Or if mother is Sahiwal, the son also be Sahiwal, right? So they will be mated afterwards, right? So in breeding always they are the same breeds, but in close breeding they are the immediate relatives. In line breeding they are relatives but not the immediate one. Maybe one of the steps are skips, or maybe one relative skip and then those are being mated. Okay. Now outbreeding. Outbreeding can be of 4 types: outcrossing, grading up, स्पीशीज हाइब्रिडाइजेशन, क्रॉस ब्रीडिंग. ठीक है. अब पहले बात करते हैं आउट क्रॉसिंग की. इन आउट क्रॉसिंग द ब्रीड्स आर सेम बट दे आर अनरिलेटेड. ठीक है. व्हाट डज इट मीन? एंड इफ यू आर गेटिंग कंफ्यूज बिटवीन क्रॉस ब्रीडिंग and then out crossing, let me highlight over here. Okay. In cross breeding they are same breed. ठीक है. same breed and related. ठीक है. In outcrossing same breed but unrelated. ठीक है. For example from Punjab we have taken the sire, maybe Sahiwal or anything. ठीक है. साहिवाल ले लेते, दियोनी ले लेते. चलो पंजाब से जैसे दियोनी सायर ले लिया. ठीक है. Then from maybe UP region we have taken one दियोनी डैम. ठीक है. दियोनी डैम ले ली. अब both are Dioni only, both are of same breed but they are not related. One is taken from Punjab, another is taken from UP. There is no relation to them. There is no, you can say, gene similarity to them. Of course देखो, don't get confused, some genes so similar होंगे because of same breed, but I hope you understand. In cross breeding it was the immediate shareness of genes but here it is not. ठीक है. तो आपके जो ऐसी ब्रीडिंग है उसको बोलते हैं आउट क्रॉसिंग. सो इन आउट क्रॉसिंग सेम ब्रीड आर देयर बट दे आर अनरिलेटेड टू ईच अदर. ठीक है. नेक्स्ट है ग्रेडिंग अप. इन ग्रेडिंग अप में क्या होता है, वन लाइक लोकल बफेलो मे बी लोकल कैटल इज टेकन. देखो व्हेन यू मूव इन द स्ट्रीट्स इन इंडिया you must have seen, right? There are a lot of cattle and buffaloes. They just roam there in the local area and you can't identify like which breed is them, right? Up isme I will take example of dogs first. You must have seen a lot of indie dogs, right? There's no such breed that you can classify it to them. We just call them indie dogs, right? The stray dogs I'm talking about, right? You know, you can visibly see a Labrador and you can say okay that's Labrador. You can visibly see for example a German Shepherd and you can just classify them. But for indie dog it is not the case. Why? Because they have been living in stray and they've been mating with like random breeds, right? So that is why we are not able to particularly classify them. Similarly the cattle and the buffaloes, they live in the open area. They might get mated with the random breeds in random progeny, right? So that is why there is no specification to the genes, and because of that random breeding a lot of time it happens that the productivity they have, it reduces. Okay. And that is why what's done in grading up, as the name suggests, we have to upgrade them. Grading up means upgrade them. So the local buffaloes or maybe local cattle, then they are mated with the superior breed so that the progeny can improve. In fact इसके लिए आपका जो राष्ट्रीय गोकुल मिशन है, दिस इज अ स्कीम, वेरी इंपॉर्टेंट स्कीम. यू मस्ट हैव स्टडीड अबाउट इट इफ यू आर प्रिपेयरिंग फॉर नाबार्ड. ठीक है. तो राष्ट्रीय गोकुल मिशन स्कीम में भी वन ऑफ द एजेंडा इज दिस ओनली, टू अपग्रेड द लोकल कैटल्स एंड आई थिंक इट्स फॉर कैटल्स ओनली, नॉट फॉर बफेलोज, राइट? बट टू अपग्रेड द लोकल ब्रीड्स ओनली so that they can be used, or the progeny can be utilized for maybe milk purpose or anything. ठीक है. तो ग्रेडिंग हम उसे बोलते हैं. इनफैक्ट आपकी बफेलो में सुनंदा आपकी एक ब्रीड है साउथ की, दैट इज मेड फ्रॉम मेटिंग विद लोकल ब्रीड ओनली. ठीक है. नेक्स्ट है आपका स्पीशीज हाइब्रिडाइजेशन. अब in out crossing, what was done in out crossing, we had the same breed, means it was cattle only, right? It was in fact of same breed also. When we say Sahiwal, both were Sahiwal. Okay, both were same breed, which was cattle. While in species hybridization, what happens here, crossing is done, ठीक है, बट क्रॉसिंग इज डन बिटवीन डिफरेंट स्पीशीज. फॉर एग्जांपल हॉर्स एंड जो आपकी डोंकी है, व्हेन दे आर क्रॉस म्यूल इज फॉर्म. ठीक है, कैटल और बफेलो के कॉम्बो से क्या बनाया गया है, कैटलो बनाया गया है. सिमिलरली थिंग हैज बीन डन इन वेजिटेबल्स आल्सो. फॉर एग्जांपल पोमैटो हैज बीन मेड बाय क्रॉसिंग पोटैटो एंड टोमेटो. तो ऐसे बनाए हैं, बट व्हाट हैपेंस इन दीज केसेस, स्पीशीज हाइब्रिडाइजेशन स्पेसिफिकली, that the progeny that is being developed, it is usually sterile, means they can't reproduce, because they have been made from different species altogether, so the reproductive organ is not that much developed. ठीक है, तो that is species hybridization. Fourth is क्रॉस ब्रीडिंग. अब क्रॉस ब्रीडिंग में क्या हुआ है, डिफरेंट ब्रीड्स आर यूज्ड. अब अगेन आई विल जस्ट कंपेयर क्रॉस क्लोज ब्रीडिंग. आई एम सॉरी, दिस वुड हैव बीन क्लोज ब्रीडिंग, आई एम सो सॉरी, यहाँ पे कंफ्यूजन हो गया होगा. क्लोज ब्रीडिंग, आउट क्रॉसिंग, एंड देन थर्ड वन इज द क्रॉस ब्रीडिंग. ठीक है, ये तीन चीजें हैं. ठीक है, अब इसमें एक बार कंपैरिजन करते हैं. इन क्लोज ब्रीडिंग द सेम ब्रीड वर देयर एंड दे वर रिलेटेड टू ईच अदर. आउट क्रॉसिंग में सेम ब्रीड वर देयर बट दे वर अनरिलेटेड. क्रॉस ब्रीडिंग में क्या है, डिफरेंट ब्रीड्स है. प्लीज कीप इन माइंड, डिफरेंट ब्रीड्स है, डिफरेंट स्पीशीज सेम है, मींस बोथ विल बी कैटल बट दे विल बी ऑफ डिफरेंट ब्रीड. ठीक है, डिड यू गेट द डिफरेंस? इन आउट क्रॉसिंग दे वर सेम ब्रीड्स, बोथ वर दियोनी, बोथ वर साहिवाल, बोथ वर रेड सिंधी, and so on. While in cross breeding, the breeds only are different. If one is Jersey, another is Sahiwal. If one is Holstein Friesian, another one is Dioni. ठीक है, that is known as cross breeding. Example, if Holstein Friesian sire is crossed with Dioni, that's a cross breeding. मूरा और सुरती का क्रॉस. ठीक है, तो this is known as your cross breeding, and this is the one that was asked in the examination because it can be of different types. ठीक है, अब types of cross breeding को देखें, तो सबसे पहले we have single two-way cross, इसको single cross भी बोलते हैं. What is this? When two breed, ठीक है, अब जैसे A और B breed है, it can be anything. Again, it can be Holstein Friesian Dioni, it can be Sahiwal Red Sindhi, any but different breeds. So when A and B breed, they are mated together, then progeny F1 is formed, which has 50% of A genes and 50% of B genes. Why 50%? Because देखो, your F1 progeny will have 100% of genes, right? 50 is carried from one parent and another 50% is carried from another parent. ठीक है, तो इसलिए 100% आपका 50% आता है. That is your single cross, या single two-way cross. Why two-way cross? Because two breeds have been used. ठीक है, next we have three-way cross. So you can understand, if two-way there were two parents, then here we have three-way cross, double cross भी बोल सकते हैं, where three parents are there. ठीक है, breed A, breed B and breed C. ठीक है, अब देखो, at one stage only two parents can be used. ठीक है, तो here A and B breeds are used, and then we have F1 progeny. Now this F1 progeny have 50% of A genes and 50% of B genes. ठीक है, now it is crossed with breed 3. अब usually in second step such breed is used of which we want more genes. How? Let's see. अब this was your F1 progeny. ठीक है, अब this F1 progeny was crossed with a new breed, that is breed 3 या breed C. Now progeny will be formed. अब 50% इस वाले it must come from F1 parent genes and 50% must come from breed 3, है ना, C breed. तो अब if you look at it in the progeny, it will have F2 progeny will have 50% of breed C, while of breed A it will have collectively 50%, right? तो if you divide further, ये 25% A का है, 25% B का है. तो in this three-way cross, what happens? Breed 3 has 50% of the genes, and then breed A and breed 2 have 50% overall. So it means individually 25%, 25%. ठीक है, next we have what? Next we have double cross or four cross. I'm sorry, यहाँ पे I mistakenly said double cross over here. That's wrong. I'm really sorry for that. ठीक है, यहाँ पे it's double cross or four-way cross. In this what happens, four breeds are used. ठीक है, it is called double cross because here four species are being used and two times like the cross is done. So here what happens, four different species are used. For example, Sahiwal, Dioni, here we have taken Jersey, here we have taken Red Sindhi. ठीक है, जैसे मान लो four different आपने कर ली. ठीक है, breed A, breed B को मिला के आपका क्या बनेगा, F1 progeny. It will have 50% of A genes, 50% of B genes. ठीक है, then यहाँ पे भी आपकी जो जो F1 progeny बनेगी, here you will have 50% of C genes, 50% of D genes, ठीक है. अब these two progeny, which is FAB and FCD, they are being crossed, and then you have ultimately F2 progeny, which has genes from all the 4 species, and means 25% from A, 25% from B. How? मतलब ये 25-25 कैसे हुआ? I hope you understand, because overall FAB will be sharing 50% of the genes and FCD will be sharing 50% of the genes, and because they were before also they were like crossbred, तो इसलिए 25% A से आएगा, 25% B से आएगा, right? And similarly 25% will come from A to C, and 25% will come from D, ठीक है. Next, coming to the next type of cross, which is known as three-way rotational cross. तो यहाँ पे भी 3 genes are required, A, B and C, but then in further stages what happens, the breeding is done in rotation. जैसे देखो, again in the first initial stage only 2 parents can be used, right? So A, B are used, and then in the second progeny C और न्यू ब्रीड इज यूज्ड. अब इसके बाद अगर आप देखो नाउ ये पैटर्न, अगेन दिस ए ब्रीड इज यूज्ड, देन बी इज यूज्ड, देन सी विल बी यूज्ड, देन ए, until and unless the breeder will get the desirable genes that he wants, ठीक है. तो if you look at it here, rotational crossing has been happened, right? A B C, A B C, A B C. That is why it is known as three-way rotational cross. Three-way because 3 parents are used, and rotational because here they are being used in rotation, ठीक है. So that is three-way cross. Next we have one type of crossing, that is back cross, ठीक है, जिसमें क्या होता है, one breed is taken B, one breed is taken A, ठीक है. A and B breeds are taken, and then F1 progeny is formed, of course, which will have 50% of A and 50% of B genes, ठीक है. Then what happens, female is taken from that F1 progeny. देखो, it's not always necessary ना that when breeding will be done only females are be formed or only males will be formed, है ना. We have to take in this next step the female progeny, ठीक है. It's very important because of various reasons. So females are easy to be mated with, and then there are specific genes that are transferred from female only. जैसे आपका डीएनए का कुछ मटेरियल है, साइटोप्लाज्मा का कुछ मटेरियल है, दैट इज ट्रांसफर्ड इनटू द प्रोजेनी फ्रॉम फीमेल ओनली. So because of these various reasons, female progeny is used, and in examination they can directly ask you also that in back cross like what is preferred, male female, ऐसे भी कुछ आ सकता है, ठीक है, because it is very exclusive thing, ठीक है. तो F1 की फीमेल प्रोजेनी लेते हैं, एंड देन यू टेक बुल फ्रॉम आइदर ब्रीड, चाहे ए की ले लो चाहे बी की ले लो. मतलब फॉर एग्जांपल Dioni was used as bull in stage 1, and here it was Sahiwal, of which the dam or the cow was taken, ठीक है. तो in the next stage we will take this particular female F1 breed, right? अब जो bull लेंगे हम दोबारा, it can be of Dioni also, it can be of Sahiwal breed also. Please keep in mind we are not saying same parent. If it would have been same parent, this would have been the category of your cross inbreeding, right? But what we are studying cross breeding. तो यहाँ पे it's not compulsory that the same parents are used, same breeds must be used, ठीक है. And then we will have the back cross progeny, ठीक है. So that is your back cross, and third we have criss crossing. इसका I could not find the picture on the internet, but it has the similar case, only A and B है, ठीक है. progeny F1 बनाएगी. अब F1 progeny में males can also be formed, right? females can also be formed. फिर इनको criss cross कराते हैं, A से B से criss cross कराते रहते हैं. That is your one of the type of क्रॉस ब्रीडिंग, ठीक है. अब लेट्स लुक एट व्हाट आर द मॉडर्न वेज, ठीक है. आर्टिफिशियल इंसेमिनेशन है, एंड वन इज एम्ब्रियो टेक्नोलॉजी ट्रांसफर. इससे पहले लाइक इफ यू वांट टू नो लाइक व्हाट आर द प्रोज एंड कॉन्स ऑफ क्रॉस ब्रीडिंग, आई थिंक इट्स अंडरस्टैंडेबल. व्हाट आर द प्रोज, वी गेट better breeds, we can also come up with new breeds, right? We get a productivity from cross breeding and all these things. And then like what are the cons, it is very expensive, right? You have to come up with such big farms where these animals can be kept, right? They taking care has to be done, and it's not always necessary that the genes the breeder desired in the further generation they actually get that, right? Because it's all randomness, right? It's all randomness, you never know. Like for example, if you look at you and your sibling also, you both are you both are different looking, although there are similarity, there are differences, but your parents are same, right? Because we don't know that which gene will become dominant in the progeny, right? So similar thing it happens over there also, not that they are only expensive and they require skills, but also you're not sure that you will get the desirable result in the progeny, right? So these were the advantages, disadvantages. Now talking about artificial insemination and embryo transfer technology. What happens in artificial insemination? So usually how mating is done, when the male covers the female and then mating is done physically, right? But now imagine, now the farmers, they would want the best quality females or cows, okay, so that they can have better productivity and they can sell it at, the milk at better rate, right? But to have such good breeds, they would require superior gene bull for to cover their cows, right, to serve their cows so that they can have better progeny. But these bulls are very expensive, right? They are in lakhs and crores, they come in lakhs and crores, good quality genes, and then you have to take care of them, you have to feed them well, right? You have to take care of each and everything, so it's very much costly and not every farmer can afford that. So here it's very, it comes as a help to be to have the concept of artificial insemination. In this what happens, so usually what happens, semen from bulls are the high quality bulls are collected, okay, and then they are just deposited into the female genital tract, ठीक है? तो अब इसके लिए भी इनफैक्ट अगर इसको मैं दोबारा मैं if I want to highlight जो राष्ट्रीय गोकुल मिशन है, in that the semen banks has also to be constructed, so these will serve as a like storage unit. So like for example, I as a farmer, I have like cows and I want them to be to be pregnant. Why? Because for two reasons. I would have more cows for milk production, and secondly, for a cow to give milk, she needs to get pregnant, right? Then only she will produce the milk. So I would go to gene banks, I will just buy the sperm, I can just or semen, I can just inseminate the females, it into the females artificially, and the process will go. So it's very much effortless, and it is also very cheaper than the traditional methods, right? So that is known as artificial insemination. Second is your embryo transfer technology. So in this what happens, for example, I have a superior cow. So superior cow when mated with superior dam has high probability of giving superior progeny, right? But one cow at a time can be pregnant at one time only, right? And that too, it might give birth to, maximum cases it might give birth to only one calf, right? Rare of the rarest cases it happens that there are two calves, but it's usually one calf, right? Because they're quite big. So now imagine for yourself, and they the gestation period in cow is around like 283 plus minus 5 days around it is, right? So imagine for yourself that if one cow will be used to create progeny, first of all it will take time, and every body can bear only certain amount of this pressure only, right? You cannot just keep on mating one cow till the death, right? Because the body also becomes weak after that. So here where the role of embryo transfer technology it comes into play. So what happens in this, here the donor cow is taken, and this donor cow is actually the one which is quite fit and has quality genes. What happens in that, first of all, super ovulation is being conducted, okay? What is super ovulation? When it is time for them to lay eggs, hormones are injected so that instead of just laying one egg, they lay multiple eggs, okay? Then what happens, these eggs are then mated with the semen, the sperms, okay? A bigger sperms are in million number in the semen, they would be able to, all these eggs they would be able to fertilized, okay? Up all these eggs are fertilized. After that, what it is formed, see, when egg and sperm they fertilize, it is known as embryo. That is why it is embryo transfer technology. So in the female body, the eggs are being first fertilized, and then what happens, then these eggs are taken out from the female body before they can implant themselves inside the female body, okay? They are taken out, and then there are recipient cows that are present, okay? Recipient cows means these are so many eggs, there has to be female bodies where these can be added so that the animals can grow, okay? So यहाँ पे क्या होगा, recipient we have females, and then these eggs are implanted inside the uterus, and the pregnancy continues, or in fact it said not only you will have better progeny in future, but along with that then these cow also, then because they are pregnant they will give birth, so they will also start giving milk, है ना? So with this, to work is done at once. Okay. And usually what happens before, there is proper stages that happens, because first of all we have to select the recipient cows, donor cows, and also the estrus cycle should also be in sync. Okay. What is this estrus cycle? So we know that all female bodies, reproductive, like the uterus, they go with a particular cycle, right? Where the first uterus, it gets all ready for the pregnancy, right? If that does not happen, it start to break down, right? Now imagine that recipient uterus is at that stage when the uterus line are going to get shedded. It will not be able to accept those embryo and implant them, right? Because now the walls are shedding. So the estrous cycle should be in sync, that the ovulation at the same time, right? And then implantation can happen properly. ठीक है, तो estrous cycle sync में होनी चाहिए. ठीक है, कलेक्शन ऑफ एम्ब्रियो, आइडेंटिफिकेशन, स्टोरेज, ट्रांसफर ऑफ एम्ब्रियोस. ठीक है, आप ये कर सकते हो. ठीक है, इसके अलावा और कहाँ पे एम्ब्रियो टेक्नोलॉजी यूज हो सकती है? अब फॉर एग्जांपल, आई एज अ फार्मर हैड वेरी average or maybe local buffalo cow I had, or average quality cow or buffalo I have. ठीक है, तो now what I would want कि चलो at this time I have this, but in progeny, in future I would want better quality cows. ठीक है, अब it's easier, or you can say cheaper. Again, it's all about the expensive and convenience. ठीक है, instead of buying a cow which will again cost in thousands, cows are like cost in thousands, while bulls are in lakhs and crores. ठीक है, some cows might be in lakhs also, but usually around in thousand you will get. ठीक है, but again quality cows will be of again lakhs. ठीक है, so it's better that maybe to just directly go to these banks, as I discussed before. Go to those banks, buy the embryo directly, and then implant it to the females in your farm, right? So then in future you will it will be cheaper way of getting like better quality cows in the future. ठीक है, तो ये कुछ आपकी new technologies, and that is all in this video. If you like the video, do let us know in the description, in the comment section, and for more such videos, subscribe to YouTube channel, and you can also join our Telegram channel to get the PDF of this and every lecture that we provide here on YouTube. Thank you everybody, and I wish you all the very best for your exam.